Looking at this split screen, it is easy to tell the running dog is alive. But what about the sleeping one? We assume it is, but if it is perfectly still, how do we know? Relying entirely on visible movement to define life is a trap. To accurately determine what is alive, we have to look closer, down to a microscopic scale. Invisible molecular movement is a strict biological requirement for life. Living organisms are highly organized structures made of tissues, cells, and smaller components. Because of environmental effects, this ordered nature is constantly threatened. If the order collapses, the organism dies. Therefore, even when an organism is entirely inactive or fast asleep, a massive network of molecules is constantly moving, repairing, and maintaining that structure. We call this continuous maintenance job life processes. It is the exact collective set of functions, running 24/7, required to prevent damage and structural breakdown in every living creature. This graphic breaks down the two primary ways organisms secure the materials for that maintenance. Autotrophs, like green plants, take in simple inorganic materials, carbon dioxide and water, and use sunlight and chlorophyll to convert them into carbohydrates, storing the excess as starch. Heterotrophs, like animals and fungi, take a different route. We consume complex substances and rely on biological catalysts, called enzymes, to break them down into simpler, usable pieces. Let's look at this step-by-step diagram to see how a single-celled heterotroph, the amoeba, gathers its food. First, it uses pseudopodia, temporary finger-like extensions of its cell surface, to reach out and fuse over a food particle. This action encloses the food, forming what is known as a food vacuole. Inside that vacuole, the complex substances are broken down into simpler ones, which then diffuse straight into the cytoplasm. Finally, the remaining undigested material is moved to the surface of the cell and thrown out into the surrounding environment. Humans process their food using a highly specialized alimentary canal. When food reaches the stomach walls secrete acid. This creates a specific low pH environment required for protein-digesting enzymes to function properly, while also killing harmful bacteria that might have been ingested. The digested food then moves into the small intestine. The inner lining here is packed with numerous finger-like projections called villi. These villi massively increase the surface area of the intestine, allowing the maximum amount of digested nutrients to be absorbed directly into a rich supply of blood vessels. Whether it is a microscopic single-celled amoeba forming a vacuole or a human absorbing nutrients through millions of villi, the end goal is identical. Nutrition is about extracting the raw carbon-based materials required for the next critical phase, generating energy. This flowchart illustrates cellular respiration. No matter what organism we are looking at, the first step is always the same. A 6-carbon glucose molecule is broken down into a 3-carbon molecule called pyruvate. This specific step always takes place in the cytoplasm of the cell. From there, the pathway splits based on the environment. In the absence of oxygen, a process called anaerobic respiration, organisms like yeast convert that pyruvate into ethanol and carbon dioxide, releasing a small amount of energy. A second anaerobic pathway can trigger right inside human muscle cells. When sudden, heavy activity causes a lack of oxygen, the pyruvate is instead converted into lactic acid. The rapid buildup of this 3-carbon lactic acid is exactly what causes muscle cramps. The final pathway relies on the continuous presence of oxygen. This aerobic respiration takes place inside the mitochondria, breaking the pyruvate down completely to yield carbon dioxide, water, and energy. Take a moment to physically pause the video right now and draw this exact chart. The breakdown of glucose across these 3 specific pathways is a highly likely board exam question. The energy released in any of these pathways is immediately used to synthesize a molecule called ATP, the primary energy currency of the cell. Noticeably, the aerobic pathway, using oxygen, yields a vastly greater amount of ATP compared to the anaerobic pathways. This schematic shows the human circulatory system. Because our bodies are large and complex, simple diffusion is insufficient to move oxygen and nutrients to every cell. We rely on a pump, the heart, and a process called double circulation, where blood travels through the heart twice during a single complete cycle through the body. Notice the distinct pathways. The right side of the heart receives deoxygenated blood and pumps it to the lungs, while the left side receives oxygen-rich blood from the lungs and pumps it to the rest of the body. This strict separation prevents the mixing of the two blood types, ensuring the highly efficient oxygen supply that mammals and birds need to maintain their body temperature. The force that this blood exerts against the walls of the vessels is called blood pressure. As a quick exam trivia point, remember that this pressure is accurately measured using an instrument called a sphygmomanometer. Plants have different body designs with a large proportion of dead cells, so their transport systems operate on different rules. Looking at this breakdown, the xylem is a dedicated tissue network responsible for moving water and minerals absorbed from the soil upwards. This movement is strictly unidirectional. Meanwhile, the phloem is responsible for transporting the actual food, the products of photosynthesis, from the leaves where they are synthesized to all other parts of the plant. Unlike the xylem, movement in the phloem is bidirectional, going wherever energy stores are needed. Whether blood is being pumped through a four-chambered heart or water is being pulled up a towering tree through xylem tubes, highly organized transport systems are a mandatory requirement. They bridge the physical gap between where energy is generated and where it is actually consumed. As chemical reactions continuously use carbon and oxygen to generate energy, they create byproducts. This text gives us our precise definition for the final life process. Excretion is the biological process involved in the filtering and removal of these harmful nitrogenous metabolic wastes from the body. In human beings, this critical filtration job is handled by the kidneys. Inside the kidneys, the fundamental filtration units doing the actual work of extracting waste from the blood are called nephrons. Plants manage their waste using different methods. They can store waste products in cellular vacuoles, accumulate them as gums and resins in old xylem tissue, or simply excrete them by shedding dead leaves. This matrix covers the core mechanics of the entire chapter. Grab a screenshot right now. If you can confidently explain the mechanisms inside these four quadrants, you have secured the knowledge required for maximum marks on this topic. Nutrition, respiration, transportation, and excretion do not operate in isolation. They form a continuous, interconnected loop. Together, they are the exact biochemical engines that keep every complex organism alive, functioning, and growing.